move right into the endocrine system. And from the endocrine, there isn't a whole lot to say here. What I really want you to focus on is this, this drawing here, this schematic. So definitely know which glands are involved in which hormones, know the axes that are involved here. So the hypothalamus is kind of the CEO in charge. Anterior pituitary is having an effect of releasing trophic hormones that can result in the endocrine glands, I'm confusing my words, sorry. The endocrine glands releasing their tertiary hormones, right? So know this hierarchy, know which ones are releasing and how that stimulates the anterior pituitary versus the inhibitory hormones, which are going to block the anterior pituitary from releasing secretions. Also think about the portal circulation. So we talked quite a bit about the portal circulation, the fact that this is where capillaries leave the hypothalamus. Instead of going back to the venous system, they go to another capillary bed, which is in the anterior pituitary. And so that results in these hormones being picked up from the hypothalamus, circulating to the anterior pituitary, dropped off, and then the trophic hormones from the anterior pituitary can then be dumped into the venous circulation and can make their way to the various tertiary endocrine glands. Okay. It's also important to think about the thyroid specifically. We talked a little bit more in depth on the thyroid gland. So this was in part 2. Okay. So think about this relay. So the fact that TSH stimulates receptors on thyroid follicle cells to uptake thyroglobulin. So thyroglobulin is a storage form of thyroid hormone that can result in T3 or T4 uptake and then subsequently being released into the bloodstream. So we talked a little bit more in detail about this mechanism, but also the feedback loops that are involved with thyroid hormone. Okay. So the short loop feedback, which is going from the pituitary to the hypothalamus, but also the long loop feedback going from the T4 being released from the thyroid back onto the hypothalamus. So these are ways that we can regulate thyroid hormone release, but this also applies for all of the hormone axes. So for example, cortisol can result in CRH release from the hypothalamus, as well as ACTH, which comes from the anterior pituitary, that can have a short loop negative feedback onto CRH as well. So know how this short loop and negative loop, short loop and long loop negative feedback can have a result on our different axes. Okay. Lastly, we spoke about the general function of the fasted versus the fed state. So it's important to think about what's happening in the fed state, the fact that we have absorption of nutrients happening, what are the main biomolecules that are being absorbed, and then we look at the fasted state and thinking about how these are now being broken down and they are liberating energy by way of metabolizing these stored biomolecules. Okay, so have more of a general sense of what's happening in the fed state and what's happening in the fasted state. Also think about the role of insulin and glucagon and know which state they are associated with, right? So insulin is a hormone of the absorptive or fed state. It's going to be trying to store nutrients up, right? It's going to be trying to store things in the adipose tissue, store proteins into muscle and the liver and so on. Whereas glucagon is a hormone of the fed state, or the fasted state, excuse me, the post-absorptive state. So this is going to be attempting to release energy, break fat down to liberate triglycerides and metabolize to make ATP that way, or it can also result in things like breaking down proteins by way of gluconeogenesis to also synthesize glucose that way. So think about these two hormones as far as which state they are aligned with. Also know which cells secrete them, right? So alpha cells secrete glucagon, beta cells secrete insulin. That is also important to know. All right. And then here's a nice summary table just to kind of summarize this. So here's a nice way to think about the individual effects of both of these hormones on your lipids, on your glucose, etc. All right. I will just emphasize here that, okay, I'm sorry. Was I not sharing my screen? Apparently not. I apologize. Okay, let's get back to this. Let's look at the muscle physiology. All right. What I was about to emphasize is that the questions from the endocrine system are going to be much fewer than the other systems, right? So prioritize the autonomic nervous system, somatic nervous system, and then the skeletal muscle. And then there will be fewer items that will be from the endocrine system and even the general anatomy of the nervous system. So definitely be guided with that information. Let's go right to the sarcomere. So let's look at the structure of the sarcomere. That's going to be probably the most important thing here. Know the hierarchy of the muscle organization. So the muscle body, the fascicle, right, which is surrounded by the epimysium, the fiber surrounded by the endomysium, and then the muscle body, epimysium, fascicle, perimysium, fiber, endomysium. So know the different connective tissue layers that are surrounding the different structures of muscle. And then we get down to the myofibril. So the myofibril, remember, these are subcellular structures. These are intracellular organelles. So the cellular unit of muscle is going to be the muscle fiber. And then inside of that muscle fiber, we have many of these repeating structures called myofibrils. Also located in the muscle fiber are things like the sarcoplasmic reticulum, which is storing calcium, very important for regulation of contractions. Lots of mitochondria, which are also important to power the myosin head for contractions, as well as the repeating units that we find on the myofibril itself, which are called the sarcomeres. So the sarcomeres are these little repeating units that are basically made of all that we see in the center here. The filamentous proteins, actin, which is in red here. So here's actin in red, myosin, which is in blue, titin, which is in green. Remember, titin is kind of anchoring the myosin heads to the Z disc. The Z disc, which are forming the periphery of the sarcomere. This is forming the I band. And the I band is going to be the lightest staining pattern formed by actin only. The A band is going to be the area of overlap. So notice there's an area where actin overlaps myosin. That is the periphery of the A band. The H zone is going to be the central aspect of the sarcomere formed by thick filaments only. And then the thick filaments are going to be anchored along the center of the sarcomere, forming the M line. Okay, so the I band, thin filaments only or actin only. The H zone, thick filaments only or myosin only. The A band is the area of overlap. Also important to know that the A band is the only area that doesn't change when the sarcomere shortens. So when you go from the relaxed state to that contracted state, the I band shortens, the H zone shortens, the A band stays the same. Okay. Also think about the cross bridges. The cross bridges are basically the connection or the contact being made by actin and myosin, and that's going to be the basis for forming the cross bridge cycling. Okay. Let's look at the states of the myosin head. So in order for us to think about cross bridge cycling, we have to review the states of the myosin head. So the myosin head has the high energy form and the low energy form. Definitely commit this to memory because you won't understand the various phases and what happens in phosphate cycling unless you associate the high energy form with having ADP and the phosphate bound. And this is where actin and myosin have a strong attraction versus the low energy form, which is when the ATP is bound, but it's not hydrolyzed, it's not cleaved. And so myosin does not have a strong affiliation. It's in fact unattracted and doesn't want to interact with the actin, okay? And so now we can think about cross-bridge cycling. So we start off with the ATP hydrolyzed. So here we're in the high energy form. The phosphate is on there, ready to give the energy to result in the power stroke. And so actin and myosin are attracted to each other and are in contact. Then the phosphate dissociates off, which is powering that power stroke. And this is where actin can drag myosin towards the M line. And then the ADP comes off, but guess what? We're still in that low energy form. Really important to think about this. Until that new ATP comes in, actin is going to stay bound tightly to that myosin because it's still in the high energy form or low energy form, excuse me. This is what we refer to as rigor. Rigor is when myosin is stuck in the low energy form and it is waiting for a new actin to come in so that it can release and then it can hydrolyze that and go back to the high energy form. So here that happens, ATP comes in, myosin and actin are unattracted to each other. And then we have the cocking of the myosin head, the hydrolyzing of that new ATP, which is returning it to the high energy form. And then now it's going to reach out and make contact with actin. And we're going to start this process again and again in order to shorten the sarcomere and bring about muscle contraction. Now, as far as the regulation, this is where we get into looking at excitation, contraction, coupling. So the regulation is really important as far as how do we turn this on and off. And in order to do that, we have to think about the structure of troponin. So troponin and the actin. So here's our actin filament. Here's our troponin complex. Binding of calcium to the troponin complex results in shifting of tropomyosin to expose the myosin binding sites. And so essentially, calcium allows for myosin to be able to access actin. When calcium is not there, myosin cannot access the binding sites in actin. So even if we did have ATP and we did have the myosin head in its high energy form, it will not make engagement with actin unless we have calcium uncovering those binding sites. Okay, so now we can think about excitation, contraction, coupling with that understanding of the actin and the regulation of calcium. So when the action potential travels down the motor neuron, crosses the neuromuscular junction here, binds to acetylcholine receptors, nicotinic receptors specifically, resulting in immediate depolarization of the motor end plate. This is going to result in an end plate potential that is going to spread throughout the muscle, specifically through T-tubules, which allow the action potential to propagate through the muscle quickly. And it's going to be resulting in the release of calcium from the sarcoplasmic reticulum by way of the receptors that are coupled on the T-tubule membrane and the sarcoplasmic reticulum membrane. Okay, so if we look at the membrane of the T-tubule, it's got two receptors that are coupling the T-tubule to the sarcoplasmic reticulum. And the sarcoplasmic reticulum forms what's called a triad. So remember we talked about this structure where we have the sarcoplasmic reticulum on one side of the membrane and on the other side of the membrane, specifically forming what's called a lateral sac. But remember, the lateral sac is just a part of the sarcoplasmic reticulum. And together, these three structures, two adjacent lateral sacs and the T tubule in the middle, is what forms that triad. So the DHP receptor, which is a voltage sensor, coupled to the ryanodine receptor, which is both a voltage sensor and a channel, allows the excitation to spread from the T tubule and will excite the ryanodine receptor, and that will result in opening of this channel so that calcium can leave the SR, which is in here or in this region, and then flood the cytoplasm and then act by binding onto troponin and then resulting in muscle contraction. Now, when we need to end the contraction, we must have the calcium ATPase pumps, which are on the membrane of the sarcoplasmic reticulum, pumping that calcium back into the SR so that we can stop or cease that muscle contraction by way of troponin dragging those tropomyosin filaments to cover up the binding sites. Okay. The last thing I want to review here is the phases of the twitch and then the forces, the types of contraction and the factors affecting force generation. So definitely review the phases of the twitch, the latent period, which is the cross bridge, the warming up. So the excitation, contraction, coupling is responsible for the latent period. The contraction phase, which is the rise in tension, this is being brought about by cross-bridge cycling, okay? And then the relaxation phase, which is being brought about by calcium pumps, removing the calcium from the sarcoplasm. So know each phase, know which one is the longest, the relaxation phase is the longest, and know the mechanism behind each of these phases. All right. Last thing here, second to last thing. Know the difference between the two types of contractions, isometric contractions versus isotonic contractions. So for isotonic contractions, the load is greater than the tension. So the load, isometric contractions here, the load is greater than tension, so the load cannot be overcome. Okay, the shortening here is a result of the tendons shortening, and not a result of the load being lifted or moved. In isotonic contractions, the load is overcome, right? The load is able to be lifted by the amount of tension being produced by the muscle. So there is a period or a point where the amount of tension levels off because it is enough to overcome the load in question. All right. And then the very final thing we want to discuss here are the factors that affect the force generation. So we spoke about this general idea that more cross-bridge cycles equals more force generation. So there are different ways that we can increase the cross-bridge cycling. One is that we can have increase in the frequency of action potentials. There were two phenomena that describe this. One is TREP. If we increase how fast we are firing action potentials, we will increase the amount of force until we have maximized the force generation for that muscle. The other is summation, the fact that because of the time constraints, because the period of a contraction is so long and the period of an action potential is so short, we could, in fact, have several action potentials repeated in the duration of a single contraction. So what ends up happening is we simply combine or sum those action potentials, which results in more force generation without relaxation happening in between, without full relaxation happening in between, okay? The second way that we can increase force generated by a muscle is basically increasing the fiber diameter. So this is basically making the muscle bigger, right? This is what happens when we work out. We increase the amount of myofibrils inside the muscle fiber. So two things are constant. We can't change the number of muscle fibers, and we can't change the number of sarcomeres on an individual myofibril. What we can do is increase more filaments or more myofibrils. And so the muscle gets bigger and it is able to generate more force. So that's the second thing that will increase force generation. The third thing is the fiber length. This one is not one that we can change in our body necessarily because our muscles are typically at a specific length, the optimal length. So this is where we have the maximum amount of overlap to result in the maximum force generation. So here's that sweet spot. Here's that ideal length where we have the most force generated for this muscle. And then the fourth thing is recruitment, along with thinking about the size principle. So recruitment and the size principle go hand in hand. Recruitment is that we can call on more fibers, more motor units to generate more force. And this is where we have the size principle in that small units with small fibers, small neurons doing small actions on small muscles are recruited first. And then large units, large neurons, large fibers doing big actions on large muscles are going to be recruited last. Okay, so the size principle governs how we can execute recruitment. All right. So that brings us to the end of important highlights for the exam.